Human survival can be considered in threes as follows. The average adult human can survive 3 weeks without food and 3 days without water, but only 3 minutes without oxygen. So why is oxygen so important? Well, oxygen is necessary for cellular respiration, which produces ATP in human cells. Remember that this ATP provides energy for your cells to function. Since cells store only a few minutes worth of ATP at a time, without oxygen to make more ATP, our cells quickly run out and can no longer function. Oxygen is the most critical molecule to take in regularly from the environment. This is because it is necessary for our cells to make sufficient levels of ATP, which provides the energy that our cells need to function. Remember that cellular respiration occurs in 3 major stages, the first of which is glycolysis. Glycolysis itself consists of 2 parts as follows: glucose activation and energy extraction. Glycolysis, which is translated as splitting sugar, since glyco means sugar and lysis means to cleave or break or split, this process splits glucose, which has 6 carbons, apart into 2 3-carbon molecules called pyruvate. Breaking the carbon bonds in glucose increases entropy and therefore releases energy to make ATP, which is an energy carrier, and NADH, which is an electron carrier. During glycolysis, glucose is broken down into 2 pyruvate molecules. The energy that is released is used to generate 2 electron carrier molecules, NADH, along with 2 energy carrier molecules, ATP. Glycolysis consists of 2 parts, glucose activation and energy extraction. Glucose activation requires 2 ATP molecules to add phosphate to generate 2 intermediate molecules. During energy extraction, these molecules are broken down further to pyruvate, releasing energy to make ATP and NADH. While 4 total ATP molecules are generated during energy extraction, the net yield of ATP overall is 2 molecules, since 2 molecules must initially be invested to start the process. Notice that glycolysis had a net yield of only 2 ATP molecules. This is much too small to sustain all of the activity of human cells. Therefore, the Krebs cycle and electron transport chain, which we discuss next, generate much more ATP. Pyruvate produced during glycolysis diffuses down its concentration gradient from the cytoplasm to the mitochondrial matrix, where 2 reactions take place. The first reaction is the formation of acetyl CoA. Pyruvate, which has 3 carbons, is broken down into carbon dioxide that has 1 carbon, and acetyl, which has 2 carbons. Coenzyme A is added to the acetyl group to generate acetyl CoA. Breaking the carbon bonds in pyruvate releases energy to make the electron carrier molecule NADH. Upon entering the mitochondrial matrix, pyruvate is converted into acetyl CoA. In step 1, carbon dioxide, which is boxed in blue, is released. In step 2, 1 molecule of NADH is formed. This is referred to as an oxidation reaction. Recall from the Unit 4 Part A lecture that oxidation refers to a molecule losing electrons. Therefore, pyruvate is being oxidized, losing electrons to donate to NAD+ to generate NADH. Finally, in step 3, acetyl CoA is generated when coenzyme A is added to the acetyl group, which is boxed in green. The second reaction that takes place in the mitochondrial matrix is the Krebs cycle, which is also called the citric-acid cycle. Krebs refers to the last name of a scientist who worked extensively on figuring out this cycle, while citric acid refers to the first intermediate product generated during this cycle. During the Krebs cycle overall, acetyl CoA, which has 2 carbons, is broken down further into 2 carbon dioxide molecules with 1 carbon each, and breaking the carbon bonds in acetyl CoA releases energy needed to make the electron carrier molecules NADH and FADH2, as well as the energy carrier molecule ATP. The citric acid cycle has several steps, each catalyzed by a specific enzyme. During the first step, acetyl CoA combines with oxaloacetate to form citrate, which is also called citric acid. The remaining steps decompose or break the citrate back into oxaloacetate, making the process a cycle. In the first step of the Krebs or citric acid cycle, the acetyl group of acetyl CoA is attached to a 4-carbon oxaloacetate molecule to form a 6-carbon citrate molecule. Citrate is broken down, releasing 2 carbon dioxide molecules and generating 3 NADH, 1 FADH2, and 1 ATP molecule for each acetyl CoA molecule. Notice at the top it states that there are 2 acetyl CoA molecules entering the Krebs cycle. That's because if we go back to the beginning, in glycolysis generated 2 pyruvate molecules, each of which were converted to acetyl CoA. Therefore, there are 2 molecules available to enter the Krebs cycle. At the very end of the cycle, oxaloacetate gets regenerated to allow the cycle to start over again. The NADH and FADH2 electron carrier molecules generated from the Krebs cycle deposit their electrons into an electron transport chain located in the mitochondrial inner membrane. The electrons move from one electron carrier molecule to the next, releasing energy with each passage along the way. This energy is used to pump hydrogen ions against a concentration gradient from the mitochondrial matrix into the intermembrane space. This concentration gradient will be used for the next step during cellular respiration to produce the most ATP so far. But right now, realize that oxygen is the final electron acceptor molecule. It combines with the electrons and hydrogen ions to form water. This is why oxygen is so important to our cells, because if we don't have oxygen, the electron transport chain will stop, and therefore the next step that we discuss will stop, and our cells will no longer be able to produce ATP. The electron transport chain is a series of electron transporters embedded in the inner mitochondrial membrane. Electrons are passed from NADH and FADH2 to several electron carriers, ultimately to molecular oxygen, which combines with the electrons and hydrogen ions to form water. As electrons are passed, energy is released that is used to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. Remember that I stated that it is the step after the electron transport chain that produces the most ATP in our cells during cellular respiration. The step after the electron transport chain is called chemiosmosis, and this occurs when hydrogen ions flow through protein channels in the inner mitochondrial membrane back down their concentration gradient from the intermembrane space back into the matrix. These protein channels contain ATP synthesizing enzymes called ATP synthase that synthesize 34 molecules of ATP per original glucose molecule. Recall that glycolysis only produced 2 ATP molecules, and the Krebs cycle only produced 2 ATP molecules as well. So before chemiosmosis, only 4 molecules are produced. So by far, chemiosmosis generates the most ATP molecules during cellular respiration. It's the flow of these hydrogen ions down their concentration gradient that provides the energy to produce ATP from ADP and phosphate. ATP synthase is a protein channel enzyme in the inner mitochondrial membrane that uses a hydrogen ion concentration gradient to form ATP from ADP and inorganic phosphate. Inorganic just refers to the fact that the phosphate is not attached to an organic molecule. When hydrogen ions flow down their concentration gradient from the intermembrane space through ATP synthase into the matrix, this provides the energy needed to make ATP. In chemiosmosis, the hydrogen ion gradient formed by the electron transport chain is used by ATP synthase to form ATP. Here we see that the electron carrier molecules that were generated by the citric acid cycle deposit their electrons into the electron transport chain. If we add up the yield of ATP from all of the cellular respiration pathways that took place, we would reach 38. This includes 2 ATP from glycolysis, 2 ATP from the Krebs cycle, and 34 ATP from chemiosmosis. However, the actual energy yield is 30 ATP molecules that are produced per glucose molecule entering into cellular respiration. The reduced yield is due to a couple different reasons. First of all, NADH that is produced by glycolysis in the cytoplasm has to be moved into the mitochondrion by active transport against a concentration gradient. This requires some ATP. It's also due to a leaky mitochondrial inner membrane, meaning that some of the hydrogen ions are able to get through the inner membrane without passing through ATP synthase channels, thus reducing the overall yield of ATP during chemiosmosis. The last point made in this lecture will not be on your exam, but is made out of interest. So the 30 ATPs yielded during cellular respiration contain 32% of the kilocalorie potential energy that was originally found in a glucose molecule. Therefore, most of the potential energy actually is lost as heat during metabolism. However, machinery generated by humans is still not as efficient as the human body itself, because if you consider an automobile only uses 20 to 25% of the potential energy contained in gasoline. The rest is lost as heat while driving.